Hello, my name is Kathryn Weinand, and today we'll be talking about defining the epigenetic landscape of dynamic pathogenic transcriptional cell states in rheumatoid arthritis, or RA. RA is an autoimmune disease that causes synovial tissue inflammation. Recent work from the AMP-RA Consortium has defined 24 RA tissue fine-grained T-cell states using transcriptional and proteomic data from within the same single cells, as seen in Figure 1. Of note, the RA pathogenic T-cell states include the TFH-TPH group, so the upper left of this UMAP, as well as the CD8 granzyme K group in this kind of lighter purple T14 cluster. The question that we then asked ourselves is, what is the epigenetic relationship between and regulation of these transcriptional cell states? To answer this question, we gathered more synovial tissue samples from both RA and OA patients to then get single-cell ATAC data from 18 samples. Again, this is just chromatin accessibility datasets, as well as some new multi-ohm datasets, of which we have 12 that has both chromatin accessibility and gene expression within the same cells. Using both of these datasets, we could then fulfill Objective 1 of characterizing the RA epigenetic cell states. We could then investigate their concordance with the known RA pathogenic cell states defined via gene and surface protein expression. Again, this is our site-seq datasets from Figure 1. This concordance is going to be investigated with a reference mapping tool called Symphony that allows us to use the multi-ohm cells that have both chromatin accessibility and gene expression within the same cells to get labels from both the ATAC epigenetic cell states as well as our site-seq cell states, mapping into the Figure 1 panel. Then, furthermore, we can fulfill Objective 3 of pinpointing the transcription factors, or TFs, regulating these epigenetic cell states, in ideally the combinations of which that are required. We could then, to do this, we needed to do some method development. We have quality controls, batch correct, and do our broad cell type clustering. Then we do our fine-grained cell type clustering. We do our Symphony reference mapping to get both, for our multi-ohm cells, labels from the epigenetic cell states and the site-seq cell states. And then finally, we do our TF inference via a dual embedding with canonical correlation analysis, or CCA, which tries to correlate these two matrices of peaks by cells and peaks by motifs, where ideally there is a particular cell state, like CTLs or cytotoxic T lymphocytes, that correspond to a specific set of peaks that also have within those same peaks the motifs that are required for CTL expression. In our results, we have defined our 5 epigenetic T cell states, 2 cytotoxic and 3 helper. So within this, we have the granzyme K cytotoxic T cells and the perforin cytotoxic T cells, as well as a CD4 IL7R group, a TFH-TPH cluster, and a Treg cluster. These were defined consistently across 30 samples, so again, both are single-cell ATAC and single-nucleus ATAC, with similar proportions across. We defined the naming scheme of these epigenetic T-cell states using promoter accessibility and gene expression of markers genes. So for example, perforin has the highest chromatin accessibility within this TA4 group that corresponds very directly to the gene expression of perforin as well. So that fulfills Objective 1. Moving on to Objective 2, we have our Symphony mapping within our multi-ohm cells to get not only the epigenetic cell state definition that we've defined in previous figures, but also the appropriate AMP-RA reference cell state as well. So we're comparing the overall reference cell type proportions and the multi-ohm cell type proportions. And we do see that they correlate strongly with a R value of 0.86. We can then note within the UMAP defined within Figure 1 where our epigenetic cell states fall, and we do see that they are grouping together with, again, all of our red cells, our our granzyme K cytotoxic cells, staying kind of clumped in the same space within this UMAP. We could then identify what cell states in the site-seq datasets those are corresponding to by doing a Fisher's exact test and defining the odds ratios for every pair of transcriptional cell states and epigenetic cell states. And we see kind of 2 major patterns within this. First and foremost, we maintain the fidelity of the AMP-RA reference T cell states. So if we look across the rows, there's usually one and only one cluster for any particular row that is red. So for example, the T5 CD4 granzyme K memory cells are only grouping within the TA0 granzyme K cytotoxic groups, and so on and so forth down the diagonal. The second major pattern is that we see multiple rows within a single column that have this has this high odds ratio of belonging together. And again, the things that are grouping on the columns are things that are all related transcriptionally. The next question then is whether or not this is just a clustering resolution error, because again, we have 5 epigenetic cell states and 24 transcriptional cell states. And to answer that question, we looked back onto the attack principal components, which are what the clusters are defined on top of. And we see a similar pattern of relatedness, where along the diagonal, we have more of the kind of less red cells, where the AUC is lower. Which means that the AUC cannot distinguish between each pair of site-seq clusters within them with a low AUC. That they are too similar that the attack principal components are unable to distinguish or unable to correctly predict which cell state of the pair a particular cell belongs to. That concludes our objective 2, and we move on to objective 3, where we have combinations of transcription factors that are corresponding or correlating to specific CVs. Within this figure here. So we do see that within the TF motifs that are along our rows here, we do see that they are corresponding differently across our top 10 CVs. For example, within CV8 here, we have RUNX3, TBX21, and EOMs correlating positively with CD8, and those are corresponding to our cytotoxic T cell states. Again, our red and pink TA0 and TA4 cell states that are correlating to the same CVs in a very similar way, suggesting that this combination of transcription factors are responsible for the regulation of our cytotoxic T cell states. So in conclusion, we have defined 5 RA epigenetic T cell states. Using the multiome cells, we can directly compare the RA epigenetic T cell states to the AMP-RA T cell states. The fact that multiple related AMP-RA T cell states were encompassed by the same epigenetic T cell state, and were still difficult to separate with attack principal components, the precursor to clustering. Again, this suggests the possibility of these epigenetic superstates, where the chromatin is plastic enough to produce multiple transcriptional cell states, may exist, likely with signals from the environmental niche and the necessary transcription factors providing further specificity. Again, this all relates back to the fact that this relationship between the transcriptional and epigenetic cell states may suggest possible treatment plans that target either the cell state, where ablating a particular pathogenic cell state that is the same epigenetically and transcriptionally may be enough to ameliorate your condition, or you may have to target exogenous factors like transcription factors or cytokines, where the transcriptional epigenetic cell states differ, and we don't want to allow a more plastic transcription, more plastic chromatin accessibility landscape to perhaps be able to produce the pathogenic cell state later on.